Good afternoon, everyone. Welcome to our seminar, From Data to Decisions, an introduction to benchmark dose modeling in public health. Thank you for joining us today. We are pleased to have you both virtually and in person as part of this exciting seminar. Before we start, let me share with you some housekeeping. Today's section will be around 40 to 45 minutes, and we will hold a Q&A session at the end of the presentation. For those joining us virtually, please use the right hand feature during the Q&A session. We will call your name, unmute you, and you can ask your question. For those attending in person, please use the microphone close to you and be sure to turn this on before you formulate your question. We do not have a chat function available today, so if you have any question after the meeting, please feel free to email us at atsdr_sss@cdc.gov. It is now my pleasure to introduce our speakers, Xiao Ji and Andrew Prusia. Dr. Xiao Ji is an Associate Service Fellow in Simulation Science Section within the Office of Innovation and Analytics. She received her PhD in Environmental Engineer from the University of Miami and completed her postdoc training at Indiana University Bloomington, where she worked on Bayesian benchmark dose modeling and toxicogenomics tool. Her work advanced benchmark dose modeling applications, including analysis of toxicogenomic data in support to public health assessment. Dr. Andrew Prusia is our Senior Service Fellow in the Simulation Science Section within the Office of Innovation and Analytics. He received his PhD in Organic Chemistry from Emory University and completed his fellowship training at the Emory Institute of Drug Development. He focused on antiviral research. In his current role, he applied computational toxicology, chemoinformatics, and machine learning models to chemical risk assessment and other public health assessment challenges. We are delighted of your participation in our today meeting. And with that, Xiao, Andrew, thank you.
Thank you for everyone taking the time to join our webinar. So as you know, like within our ATSDR's public health assessment, there are several layers of data that will be collected and will be analyzed, such as environmental sampling data, exposure data, health effects data, and the community concern data. There is one key information, is toxicological effects data. So, like for the toxical profile, it gives you the information about the potential health effects and also the health guidance value or the MRL value, the minimum risk level. So you can compare with the environmental exposure level to determine the potential risk and answer some key questions, such as whether there will be some potential health effects. Is this exposure level safe to the community? So today we will walk you through how the benchmark dose modeling has been applied to derive those MRL, those health guidance value, and how can use BMD modeling to help your public health assessment.
So we're going to switch back and forth a little, so just to keep it interesting, I guess. But I want to give you a brief history of risk assessment. To my knowledge. Going back, you can think back to 1983, the NRC, National Research Council, they published the Red Book. It kind of codified a lot of the processes in chemical risk assessment. And then not that much later, the benchmark dose concept, which we'll talk much more later, was first introduced in 1984 by Kenneth Krupp. And this, it took a few years, but it developed a lot of interest, and ultimately the EPA developed their own software to do this, to make it easier for the risk assessor to do it on their own. And this was their BMD gas software, is released in 2000. Then a few years later, the National Research Council is recommending to quantify uncertainty in chemical risk assessment, and that's a process we're still undergoing. A few years later, Bayesian approaches, Bayesian modeling is first applied in 2014 to benchmark dose, and we'll talk more about that later. And looking to the future, just recently, you may have seen that the EPA has a target for eliminating all mammalian animal testing in 2035. So that's going to be coming. It's going to be here very soon, and it's going to change the kind of studies that we have to develop risk assessments, and it's going to impact how we can do environmental public health. So if we think about I kind of split it into these two sides. We have the 20th century assessment. It was very important, established the essential procedures that we use today, really used to some great public health effect for legacy chemicals. I'm thinking dioxins, organophosphates, others that you guys know about very well. It's really dependent on animal testing for almost all of these assessments, usually in rats or in mice. Sometimes other mammals. And we're using these LOEL and NOAEL concepts. So this is the lowest observed adverse effect and the no observed adverse effect level that we're seeing in an animal toxicity study. And after you get those points of departure, the LOEL or the NOAEL, then the standard procedure is to apply a fixed uncertainty factor, usually 10, to take that point of departure and then derive a human health guidance value. For ATSDR, that's our minimal risk level. For EPA, that would be their reference dose or reference concentration. And so that is the 20th century. And then we're thinking in this century, we want to apply increasingly quantitative approaches. We want to utilize animal data where we have it. We want to use it as most effectively as we can. We definitely don't want to redo experiments. If the experiment has already been done in animals, we want to utilize that animal data as effectively as possible. And we want to be able to use non-animal data. We could use epi studies in humans. That actually eliminates a lot of uncertainty because now you're in humans. Or we can use toxicogenomics, and Cha will talk later about how that can actually get to the mechanism of the toxicity that you're seeing. And like NRC proposed back in 2009, we want to use probabilistic and population-based assessment methods. And there will be examples where we need to apply these methods to complex materials.
So the foundation, as you know, like for toxicology, it's a dose-response relationship. So we can say like everything is toxic and it depends on the dose level. So the dose-response relationship, it tells you the relationship between the dose or the exposure and some measure of the biological significant effects. So here I'm just showing you like a plot for the potential effects. So as you can see, as the dose increase and gradually it will occur the first critical effects. So that one is the most sensitive one. And as the dose keep increasing, it will have other kind of effects for different organs, such as the neurological, immunological, or the liver tox. But for the health guidance value, we rely on the critical effects to derive the MRL or other kind of health effects. So like when you already have identified the study and the critical effects, dose response shape, the key step is to find the point of departure. which is a turning point on the dose response curve that will making the beginning of non-cancer effects. So we are assuming below this turning point, there is minimum risk, and above this dose point, it will have some potential health effects. So as you know, for the MRL development, most of their study are the animal-based. So how to translate the animal-based results to human? We need to typically apply two types of uncertainty factor. One is animal to human, and that is typically 10, a factor of 10. And when you have information for the dosimetry adjustment, like you have the body weight for the rat and you have the body weight for the human, you can adjust that part. And this is part of the animal to human uncertainty. When you have the dosimetry adjustment, you can apply a lower uncertainty factor for the animal to human. That's 3. And the other uncertainty factor we use is human variability, and usually it uses a factor of 10. So the process is when you have the point of departure, it can be either NOAEL or the BMDL value. BMDL is the lower confidence interval, 95% interval of the BMD value. And then you divided those uncertainty factor, you will get the MRL value, which is the level of a chemical a person exposed to without a detectable health risks. So far, ATSDR has published 484 MRLs, and this can be grouped by different exposure durations, from acute, which is less than 2 weeks, intermediate is less than 1 year, chronic is longer than 1 year. And it also can group by different exposure routes. For inhalation, we have like 175 MRLs. Orals, we have 305 and 2 for the radiation.
Okay, so now I'm going to walk you guys through using the NOAEL approach to do a point of departure and how that works. So if you're looking here, over here on the left, we have what you can imagine would be a typical result you would get from an animal toxicology experiment. And you have 4 dose groups where you dose the animal at different levels, and then you have a control group where there's obviously no dose. And then you have responses. So we're looking at responses going from 0 to 1 or 0 to 100%, right? So what you can see here is like there's a dose response. As you increase the dose, you increase the response. And the way you determine the NOAEL and LOEL is the NOAEL is the highest dose at which there was not an observed toxic or adverse effect. And the way you determine that is by comparing it to the control group. So you're trying to see if there's a difference from the control group using this statistical test. Now the LOEL, that's where you actually can tell a difference from the control group. And you see the error bars here are based on the kind of experiment you have. In this case, it's dichotomous data. So what if you guys know a lot of statistics, you might be thinking about what that statistical test would mean, and you probably have already thought that the sample size would definitely affect the NOAEL-LOEL calculation. You'd be right. So for each dose group we have on the left, there's 50 animals in each dose group. So we have 250 animals in this study. And you can see with that level of statistical power, you can determine that this dose in the middle at 100 is the LOEL because it looks different from the control group with a p-value less than 0.05, right? Now the dosing at 50 looks exactly the same, so that's where the NOAEL is. So if you have the same, imagine doing the same experiment, but now you've reduced the number of animals, same chemical, same lab, same conditions, everything's the same except you have taken it, each dose group now has 10 animals instead of 50. how would that affect this calculation? So decreasing to 10 and everything else is staying the same. Now, because of the statistical test you've done, you actually cannot see a difference between this dose at 100. So the p-value is actually 0.1, a little over 0.1. So you can't actually say that that's different from the control group. So that becomes the NOAEL in this example. And the NOAEL at 150, that is less, the p-value is less than 0.05 is typical, the typical alpha level. And that, so the NOAEL becomes 150. So you can see that it makes a difference how the experiment is designed and how many animals are in the experiment, even if the underlying toxicology is staying the same. So these are the limitations of the NOAEL approach. So sample size, the ability to detect a treatment response, it decreases as the sample size decreases. So if less animals means your NOAEL is going to be higher, less protective. The dose selection. So your point of departure can only be the actual doses that were given to the animals. If the effect starts happening between doses, that's not part of the dose response that you can use. And then the shape of the dose response curve, if it's very steep or very shallow, that's not taken into consideration. You're just doing this pairwise comparison from the dose group to the control. It doesn't tell you anything about how quickly it responds, how fast the dose response goes up. And then there's many times when we're doing the risk assessment and we don't have a NOAEL. So what that means is we have the control group, and then the lowest dose is where the effect is actually significantly different from the control group. So then we have to use that NOAEL as the point of departure. And of course, we want to be protective, and we know there's effects at the NOAEL. So that requires an additional uncertainty factor in our process of using a factor of 10 for using the NOAEL. And then the other issue we have is when you're trying to decide which is the most sensitive effect, like Chao was showing with the critical effect, you may need to be comparing lots of different studies, or even within the same study, they measured many different endpoints. So you need to be able to compare which study is the most sensitive, and they might not have used the same dosing as the one study. So the direct comparisons are difficult.
So like Andy said before, like we transit to the 21st century risk assessment. So instead of using just the statistic test to determine which dose is NOAEL or NOAEL, here we transit to use benchmark dose modeling to model all the dose response data and to model all the dose response curve to give you a shape for all the dose response relationship. So the benchmark dose modeling approach is a statistical method to describe the dose response relationship. It can give you the whole information for all the dose group you have tested. There is one key parameter you need to define is a BMR value. So it's a change in response for an effects related to the background response. So you see here, I have the BMR as a 10% increase compared to the background level. And the dose related to the BMR change is the benchmark dose level. So the blue one here. As you know, for the experiments, for each dose level, there are like variety and uncertainty from the response for different animals. So for each dose point, we can assume it's a distribution, and it can be either normal or log normal distribution. So here I'm showing you a normal distribution for one dose level, and the BMDL is a lower confidence interval of the distribution of the 95% confidence interval. And the BMD upper is the upper level for the 95% interval. So when you like the figure here. So the blue dashed area is the uncertainty for the BMD estimates. And the red line here is the BMDL value, is the lower confidence interval of the 95% confidence interval of the BMD distribution. So when we using the same data set that Andy described before, like we have the same incident rate, but we just have different dose group. So on the left here, you can see like we have the 50 animal group for each dose level. This one is for the 10. So when you do the statistical testing using Fisher test, it will give you different value for NOAEL and LOEL. But for the BMD method, the BMD value is very stable. So you see here, they are like all around 80. But for the BMD, when you have the same BMR, like the left figure, it has less uncertainty. As you see, the blue area is smaller than the right figure here. That makes sense because for the left one, you have more animal for each dose group. So those uncertainty are controlled by those study design. And here, when you have less animal group, the BMDL value is lower than the left one. So you have the BMDL value. So consider the uncertainty for the study design.
So you may already be thinking of some of these advantages that you get from the BMD approach, and the obvious one is the sample size, as it appropriately considers the number of animals in each dose group. So if the number of animals decreases, the uncertainty for the benchmark dose will also increase. So you'll get a lower BMDL, more protective BMDL. The other thing in dose response, dose selection, you're not constrained by the dose actually used in the study. You can use any point along the graph, along the curve. You can use that as a point of departure if appropriate. You've also, instead of just doing this pairwise comparison where you ignore basically all of the dose response except the control and the treatment group, you're actually utilizing the full shape of the dose response curve. So remembering that each dose group is a lot of animals who gave their lives for this study, right? So you're using that information as much as possible. And if there's a NOAEL missing from the study, you can still, that's no issue. You can still calculate based on what you define as the benchmark response. You can still calculate a BMD and a BMDL, even when a NOAEL is missing from the study. And you can just compare, it makes it easier to compare against different studies that use different dose groups, because you can determine what the appropriate BMR is for each study, and then compare the BMDs as a point, as your point of comparison.
So let's see, like what kind of data you can use for doing the BMD modeling. So for the traditional way is to use the animal-based toxicology, is using the in vivo animal testing. But the animal testing, there are like several disadvantages. Like first, as you know, like from the acute to chronic, it usually takes several years to complete. So chronic, at least 2 years. And as you know, there are like thousands of chemicals in the market. And EPA, every year, they have like thousands of chemicals they need to regulate. So if you only rely on the animal toxicity data, it doesn't have the capability to evaluate for all the chemicals in the market. So when you don't have the MRL value for like those chemicals you identified in the environment, there are also other kind of data you can apply for the benchmark dose modeling and get a health guidance value. Those data could be the EPI study. So when you have different exposure groups and the odds ratio or the risk ratio, you can use the benchmark dose modeling to derive the health, the safety health guidance value. And the other method is the new approach methods. As you know, the risk assessment has been transit from the animal-based study to like more human-related study. So for the animal effects you observed in those animals, are they for sure will appear in the human? There are like lots of uncertainty there. If we, instead of using animal data, we use more human-related data, so that will reduce the uncertainty there. And also for the like new approach method is more cost efficient. Instead of doing the experiment for like several years, you can get the response at the gene level or at the cell level within several days. And this can be applied to like some emergency response, like something you need the data more efficient. So today we will give you like 3 case studies for each type of data and walk you through like the benchmark dose modeling process. Here is just like a summary of how the BMD will work. So first, you need to have the appropriate toxicology data. For animals, it can be either dichotomous or continuous data. Dichotomous is a kind of binary data that you like either the animal have the tumor or not. For continuous is like, for example, the liver weight change for the animals. And the other type of data is the epi data. You need to have several exposure group, and then you calculate the odds ratio or the relative risk ratio. For the NAM data, you also need to have the dose response data. So here I'm just like showing you like what kind of model you can use for different type of data. So the one in the green box, those are for the dichotomous data. And in the orange is for the continuous data. And you also need to define your BMR value. So whether you think like 10% will be a safety change compared to the background level or like 1 standard deviation, you need to decide the BMR value. And with all these choices and inputs, and then the like some kind of software, like the BMD software, like will give you the BMD estimates, and you can use it for the health guidance value. So although we have like different type of data, but the goal is the same. We want to use this data to determine the chemical exposure limits that can protect public health, that can protect human health.
Okay, so I'm going to walk through kind of your typical toxicology experiment that we did. We actually recently did this for the MRL for Xylene when we went through the risk assessment. So we have in vivo data for an oral Xylene exposure. So the study we picked was a 28-day repeated dose toxicity study with Xylene dosed by gavage in rats. And so what they measured was, well, of all the things they measured, the most sensitive effect appeared to be a neurological effect. And this was an increase in locomotor activity. So the rats were kind of hyperactive, actually. So looking down at the table at the bottom, you can see there appears to be a dose response. And then there's it's graphed. The table on the bottom left is graphed on the bottom right. So you see there appears to be a clear dose response. We go from the control group all the way to 1,000 milligrams per kilogram per day in the highest dose group. And that dose at 300, it's different based on the statistical test to the control group. So that's defined as the NOAEL. And that dose at 100 doesn't appear to be any different at all from the control group based on what the animal behavior. So you can take this data. And well, one thing you see from this data is that there's only 5 animals in each dose group. So even less than we talked about before, where we had 50 and 10. Now we're down to 5 animals. And because of that, because we're measuring the average result, the average activity from each animal, there's some wide standard deviations for each dose group. So we get the mean and we get the standard deviation, but there's quite a bit of change and quite a bit of variation. You can see that in the graph on the bottom right. So what can we do with this data? Using that software that was developed by the EPA, benchmark dose software, way back in 2000, there's been several versions now in version 3.3 or something like that. And it's online, it's available free. And what it does is uses a maximum likelihood method to fit a panel of models to the data. So it's a web-based interface. You enter the dose response data in a format like this, just telling you how many doses, what the doses were, number of animals, the mean of the responses, and the standard deviation. And so the next step is, like Chow mentioned, very important, we're going to define the benchmark response as one standard deviation away from the control group. So we get to select what that benchmark response is. It could be, if you didn't want to use one standard deviation, you may want to use something like 10% change from the control group, or you could use absolute difference, or if there's liver weights or something. So you have a variety of options here based on what you know about the toxicology in the animal. In this case, one of the easiest ones is just to use one standard deviation. So you're just looking at where you're able to detect a difference from the control group. So here we fit the panel of models, and it actually goes through some logic, and it selects what it thinks is the best model for you, which is in this case the linear model. So this is giving us a good fit, and it has a low number of parameters, the fewest number of parameters. And so the benchmark dose would be the dot you see here in the middle here, which is 305.6. And of course, there's a confidence interval for that benchmark dose from 219 to 495. So we're going to use that lower limit at 219. That's going to be the lower limit to our benchmark dose. And that's going to be the point of departure that will carry forward to do the risk assessment. Yeah. So even though the data, is not the best, there's only 4 doses, including the control, so only 3 actual doses. We only have 5 animals. There's a lot of variability in the measurement. The result is still reasonable and not 2 times overprotective, which it would be if we had used that NOAEL. So what I mean by that is if we go through the whole process of getting the MRL, we take the BMDL, we divide by the uncertainty factor from animal to human and the uncertainty factor for human variability. We get to the BMDL-based MRL of 2.2, right? If we had to take that NOAEL, which was at 100, we would divide it by the same uncertainty factors, and the MRL would be 1. So you can see how we're actually closer to what the data is actually telling us, and we're not overprotective, which being overprotective, it does carry a cost to it if you're dealing with a cleanup or with a public health issue.
So the second case study we want to share with you is to use the EPI data for the health guidance value derivation. Like for this is arsenic exposure and the blood cancer. Although at ATSDR, we do not do the cancer risk assessment, like EPA, IRS will do that. But this is just like an example with a published study that has used the benchmark dose modeling. And if you have like similar EPI data, you can just apply the similar concepts to your data. This is just like an example. So for this study, we use a published EPI study from Chen, 2010, and it's in South Taiwan. And the resident has consumed the water from their private wells that contains the arsenic pollution since late 1940s, and they have the clean water until 1990s. So for a long time, those residents has been exposed for the arsenic. And for this study, they interviewed over like 8,000 residents, and they measured the concentration in their wells. So here you see they grouped the detected water concentration into 5 exposure groups. And they also report the cases and non-cases for the blood cancer. So we like the study. There are like some published way that you can adjust the range of exposure into a midpoint. So that's the dose level for each exposure group, and then you can use logistic regression to calculate the risk ratio and also its confidence interval. As you can see here, that's the response. There is a clear dose response relationship. As you see the dose increase, the risk ratio increase from 1 to around 8. So that's a lot increase. So here is the BMD modeling results. When you define the BMR as 1% of the increase of the risk ratio, so that's the BMD and BMDL value you will get is around 10. When you have the risk ratio at 0.1 or the 0.05, this value is also around 10, which means the method is quite stable. But when you use the NOAEL or LOEL approach to determine the point of departure, so you see the NOAEL here is around 144. That will much underestimate the risk. And for this value derived based on the exposure level and the EPI study, and this is also agrees with the current EPA's guidance value for the arsenic in drinking water, which means this value is quite protective for the public health. So and then we will move to the other case studies for using the NAMs. So as we mentioned before, for the traditional toxicology test, it usually takes a long time, at least 2 years to complete. It also measures the change at the organ or the tissue level. So, you know, for the disease, instead of at the gene level, it takes time to develop to be observed at the organ level. So that makes sense. It takes time. So like in 2018, the NTP, the National Toxicology Program, they published a guidance on how to use the benchmark dose modeling for the genomics data. In general, there are 2 types of NAM that you can use. One is the in vitro method. The in vitro is measure like the cell changes. The in vivo is still use animal, but it measures the response of the genes at the gene level instead of at the organ or the tissue level. And studies have shown that the transcriptomic-based point of departure from the short term, like within several days, they are highly correlated with the empirical point of departure from the long-term exposure. Those work are from like EPA's group and also from the NIEHS group, like lots of like current other agencies, they are adopting to the toxicogenomics way. So for the in vitro method is kind of different for the in vivo. So as you like measure the human cell response, you need to have a IVIVE or the in vitro to in vivo extrapolation to translate the results to the human related value. That's a different kind of uncertainty factor compared to the traditional animal-based study. As you know, we discussed like the toxicogenomics, the method, they can give you response within several days and is more cost efficient instead of like testing those animals for like over 2 years, and then you have uncertainty for like those experimental designs. And what if at first you have 50 rats, and at the end you only have 10 left? There are like lots of uncertainty that you cannot control for a long period of time. But for the toxicogenomics, it can provide you the solution to like reduce the animal kills and also more efficient to evaluate toxicity. And also, since you know, for the disease development is from the molecular level to tissue and then to organ to the population level. So if we know what will happen at the molecular level, we could know more about the mechanism of like the potential pathway for those exposure and toxicity. So the toxicogenomics data also can provide you some mechanistic explanation. As it's more cost efficient, it has been applied for the emergency situations. Back in 2014, in the Elk River chemical spill, the NTP, the National Toxicology Program, they have conducted extensive toxicogenomics to measure the potential toxicity. As back at that time, they don't have the toxical information for this chemical. So here is to show you like how the toxicogenomics data looks like. It's like similar as the other dose response data. So like you have different genes, but for like one microarray, when you measure, you can measure thousands of genes. You have the control level and you have different dose group. So for each gene level, you can treat it as a continuous data, and then you use the benchmark dose modeling to get the point of departure for this gene level. And then the gene at the gene level can further grouped into different biological pathway. Like for example here, this pathway is a cellular response to the chemicals, and this one is related to the tumor incident. So you can have some like information like what will happen at the molecular level and how the adverse outcome will develop from different level. This is like just a coloration to show you like how the toxicogenomics data derived point of departure is correlated with the ambiguous point of departure. It was from one of my previous publication. So they are highly correlated. The R score is like over 0.8.
So far we've talked about how to handle the derivation of a point of departure. So that being either from an epi study or the NAM or from a tox study. But what we haven't talked about so far, we've foreshadowed, I guess, the uncertainty factors. So this is another level of uncertainty that we're getting in our final risk assessment in our MRL. And as Chao was saying, we have to go from an animal to human uncertainty factor, and then we have to account for the variability in humans. We're very different. So typically, as Chao mentioned, there's an uncertainty factor from going from animal to human. Sometimes you can have a dosimetric adjustment, which would be based on the physiology of the animal, particularly if it's an inhalation exposure. You can adjust based on the rat anatomy being different from human. So that if you can do that, that's great. And then you can use a reduced animal to human uncertainty factor, usually 3, because you've accounted for that difference between animals and humans. So that's great, but you still have to do the human variability. And that has been, that's a factor 10 is what we use typically, and that's assumed to be conservative, but it's unclear by how much, and it's unspecified as to what population fraction of that, of humans you would be covered by that. And this factor of 10 goes all the way back to 1954, first done by the FDA and their risk assessments. So it is a data-based number, but it hasn't been changed for quite well. And so we want to think about how we can quantify uncertainty a little more data-driven. So if we have an MRL, we're assuming it's going to be protective, but because we're using these fixed values for uncertainty factors, 10, 10, or 3, it's unclear by how much, and it's not specified what fraction of the population we're covering. So if you think about this, if we have a site and there is a real exposure above what the MRL dose is, what does that mean? How much of the population will be affected? Do we expect 50% of the population to have health effects? Do we expect 10%, 5%? The MRL as it stands now doesn't really tell us that answer. And then the question is, what if it's a little bit lower than the MRL? Does that mean there's no risk at all? Well, there's a lot of variability in the human population, a lot of variability in how we do the animal to human. So that's an open question. So some academic groups and the WHO have published guidance where we actually go beyond these fixed uncertainty values, beyond the MRL or beyond the reference dose, and use a concept called the target human dose.
So this is like the recommend approach. So here, as Andy discussed, like for the MRL, you have a fixed value for the point of departure, and you have fixed value for the dosimetry adjustment, and like either 3 or 10 for those uncertainty factor. So those factors, they are the same for all the chemicals and for all the MRL derivation. So instead of like for the WHO's recommend approach, instead of for each variable, instead of you having a fixed value, you have a distribution for each parameter. Like for example, the benchmark dose, you have a distribution, and for the dosimetry, you have a distribution, and this value can be obtained based on the historical toxicity data. And also for the uncertainty factor, you can like use like the PPPK model or some like clinical data, like the pharmaceutical data, to access how the human variability could be. So like the group at Dr. Wishwei Chu, he is a professor at Texas A&M. Like their group has done like lots of work on quantifying like what's the distribution for each parameter could be. So like if we change this equation to the health dose, and instead of getting a fixed value for the MRL, you will get a distribution for the health dose value. So here is a figure to show you like the dark area is the 90% percentile for the estimation, like when you have a fixed effects, like let's say 5% as the BMR. And here is 95%, and then the other one is 99%. So like in the real public health assessment, when you have a certain dose level, it will tell you what's the probability of how much percentile of population will be affected. Like the dots here is represent the lower confidence interval for the 95% is like 1. So you can say there is 5% of chance for the population level, they'll have like 1% of the incident rate. But if you change the measurements in the public health assessment, you can get a different estimate for the probability and how much people will be affected. So instead of a fixed value and unknown risk factor, you could use like the distribution to quantify like how much risk and what the probability would be?
So this is our last slide. So conclusions, we're thinking that environmental public health, it's moving to a more data-driven framework. It was always driven by data, but there's going to be more quantitative approaches applied to it. And thinking about, over the past, there have been amazing wins in public health, environmental public health. The chart on the right is a paper that just released. It's the lead concentration in archived hair samples from Salt Lake City residents. You can see this amazing drop over time through the actions of ATSDR and EPA and others. And that's a huge win. That has huge public health implications for the entire nation. So these kind of wins are going to be more difficult in the future, I think. But what we do have are more powerful analytical biomonitoring, EPI, and GIS tools. And we can apply those to this framework, to public health. And reducing animal testing will also will require us to use new approaches, analysis, and interpretation. And it's clear that benchmark dose modeling will be a key component of this. And I think that our agency is well positioned to navigate this new landscape. And that's because we have so much expertise in these areas. We're a multidisciplinary agency. And we have expertise in communications and policy, and those are essential. So I think I'm optimistic for the future, and I think that we can be a part of this more quantitative approach. And with that, I think we're ready for any questions. Oh, okay, we have a question in the audience.
The difference between benchmark dose and the other one is that it doesn't seem to incorporate variance so much as the mean. And then so you go from 10 samples to 5. What is your floor?
Yeah, so you have to have some number of animals. So probably 5 is about the minimum if you're doing an animal-based study. If you have an epi-based study, it could be different, right? Because you could have a lot more people enrolled in that. So you need to be able to detect the difference, and really, in order to get, because you have to know that variance for each dose group, really 5 is kind of the bare minimum. So, but that's typically, for mice and rats at least, that's usually the minimum of animals in each dose group.
Curious, just kind of outside of my field, so I'm just being curious about, so if you have say you have an MRL from a known toxicant and maybe something that's well studied, you've got an MRL and you go in and you do the modeling on the BMDL, and you get, your MRL down here, do better quantitative modeling, and you get a value that's maybe more reasonable, not based on just dividing by 100, right? Seems more reasonable. Are you aware of any studies where they go back in, maybe in vivo or with genetics toxicity assays to ground truth that the difference between the two and test the accuracy of the modeling?
Yeah, like in the Europe groups and also the US group, there are lots of technical report to compare like the previous method, like the NOAEL, LOEL, and the benchmark dose modeling. So EFSA, like the Europe Food Safety Association, they published the guidance on like transit from the NOAEL and LOEL to the benchmark dose modeling. They have done like lots of assessment for this study. And then for the toxicogenomics, NTP, the National Toxical Program, they also conduct extensive testing for those toxicogenomics data, how their value are correlated with animal-based study. That's like those methods, they are very well studied, I would say. especially for the benchmark dose, I think is nowadays well accepted one. For the toxicogenomics, maybe still will like take several years to like be more systematically be used for the risk assessment. But I know the NIEHS group, they are working on that. They have developed a model for that. So and there are like some workshops like for those areas. That's and also like the FDA, NIH, like last year April, they announced they will not support the animal testing. So what's the alternative? That will be like I think in the future we have to use those more NAMS data.
Yeah, just to add that, I'm thinking of a paper from EPA where they correlated different, they did this exercise. They looked at the correlation between BMDLs and NOAELs, NOAELs in this case. And they were very high correlated, like 0.9 or some higher, but there are examples where there was pretty big differences. So there are cases where the overall correlation was high, but there are some outliers in that regression that were pretty high that would change what you would do on the ground basically based on the assessment. So if you're online and you want to ask a question, we should be able to see it if you raise your hand. If not, that's okay. And if you guys have questions that you want to submit in writing, please use the email address on the screen. Okay. With that, I think we can dismiss. Do you have anything else? Thank you for your attention. Thank you for coming to this lunch and learn.
Thank you, everyone.